we have gone through the structure of DNA, right? Went through the structure and how double helix, anti-parallel, the base pairs, the nucleotides, etc. We went over the structure and related that to its function in terms of replication. Okay? The fact that one strand, the old DNA, serves as a template based upon base pairing rules. Okay, so that for replication and the formation of new strands of DNA, which then passed on to daughter cells. Okay, we went through the ins and outs of DNA replication. So, enzymes that are involved in DNA replication include what? What enzymes are necessary? What proteins are necessary for DNA replication? Helicase. And what does helicase do? It unwinds. Okay, give me another protein. Topoisomerase. What does topoisomerase do?
Relieves the tension.
Relieves the tension ahead of helicase. Okay, Abby. Primase. What does primase do?
Puts down the 3' RNA.
So it puts down the short RNA primer that provides what specifically? What specific part of structure is necessary? That 3', right? That 3' OH on the RNA. Okay? And that 3' OH on the RNA is the starting point for what protein? What next? Polymerase. All right, DNA polymerase, which will come in and start synthesizing new DNA. Is there anything else? Ligase. What is ligase?
It fills in the...
It joins. It joins unattached DNA parts. So it joins the parts. Why is ligase important in joining the parts? Why is it necessary? Fill in gaps that are present from what?
The RNA.
From the RNA primer. What else? So that what is produced in fragments? Which strand is produced in fragments? The lagging strand. And why do we have a lagging strand and a leading strand? What's the difference between the two? All right, so directionality. One's always moving towards the replication fork. One's moving away. Okay? And what about the continuity? Which one is continuous? Which one is discontinuous? The leading strand is continuous, meaning that it happens in one complete step, while the discontinuous lagging strand is broken up here and there. Okay? Any other proteins that we didn't talk about, that I didn't mention, that are important? Single strand binding proteins. And what's their function, their role? They bind to the single-stranded DNA and they keep what? The fork open. And where does replication begin? At an origin. Okay? At the origin. So we've gone through the structure of DNA replication. We also went through how DNA is repaired, right? Nucleotide excision repair, sort of taking nucleotides out or a chunk of nucleotides out and then fill it back in with DNA polymerase. We also talked about mismatch repair, where one nucleotide can be taken out, and that's based upon what? How does the cell recognize that there's a problem with DNA base pairing? Proofreading. And how does it recognize it through proofreading? What is the signal? Why can it recognize that A to T is okay, but T to T is not?
The width of the strand.
The structure, right? The structure is changed. So the diameter from one strand to another is changed when you have two purines or two pyrimidines stuck in as part of the base pairs. All right, questions up to there? All right, so we went through structure and function of DNA. And now today what we're going to go through is sort of how it's used. All right, and last week in lab, you did a cloning procedure. Right? You took a piece of DNA, you did a transformation. Right? We talked about what transformation is, where bacteria picks up outside DNA and then it can express it. Okay? You did that. And from the results from lab, what I've seen, about 90% of the groups, maybe 95% of the groups, actually got transformation and got to produce a DNA that, or a bacteria that glows under UV radiation. All right, meaning that that bacteria is going to produce or express whatever genes are on the DNA that it picked up. Okay? Today we're going to talk about what you're going to do in lab this week, all right? And also how DNA is used for other scientific stuff. All right? Other scientific procedures that we can use for DNA. All right? And so part of this is based completely upon the properties of DNA. Okay? First off, we know that DNA, the strands are complementary to one another, right? Meaning that the sequence on one strand, ATCG, CGA, okay, is complementary to the other, right? So that, A to T, T to A, C to G, etc. Those complementary base pair rules that were set forth by Chargraph, all right, are still, we use that to our advantage. That's a property of DNA that we can use. And one thing we can do is what's called nucleic acid hybridization. So the property or the basis of this nucleic acid hybridization is that you may have a stretch of a single-stranded piece of DNA. Is that the way that the DNA wants to be in a single strand? Yes or no? No. It wants to be what? Double-stranded. And what is that other strand going to be? It has to be what? Complementary. So if you know a strand of DNA is ATCGCG, whatever it might be, you can predict what the opposite strand is going to be. Okay? That complementary base pair rules and the complementarity of DNA have been used by scientists in a variety of ways in manipulating DNA structure, genes, genetically modified organisms, which is big in the news now. Right? This is all based, the basis for this lies upon the complementarity of DNA. Okay? And so we can get into sort of that what we call hybridization, forming one or two strands together, a hybrid between two strands. That forms the basis of what genetic engineering is. All right, and we're going to get into some aspects of that. All right, so first, DNA cloning. All right, so you may have heard of cloning, okay, as in replicating organisms. Well, cloning was first talked about in terms of DNA. All right, and cloning pieces of DNA, making more copies of a gene expressed in an individual, in an organism, etc. All right? So, at first, scientists wanted to work with specific genes. They prepared well-defined segments of DNA in identical copies, a process called DNA cloning. All right? And we'll go over sort of the background of this in a second. All right? And this is what you basically did last week in lab. You took a piece of DNA and you mixed it with bacteria. Okay? That DNA was a plasmid, a small piece of circular DNA that actually got into and was incorporated into the bacteria. All right? And on that plasmid was a gene. So it looks something like this. Here's your plasmid. right? And on that, there's a couple genes on that plasmid. This was your, I don't know what it was called, the P-glow gene or something like that. And then also over here, it had a ampicillin resistance gene, whatever it might be, right? That look familiar to you? The ampicillin was for selection, so that you could say that this bacteria picked up the DNA and is expressing it, while other bacteria that died from the antibiotic did not. All right? And so, but what you could do is you could take that plasmid that you used is really a result of DNA engineering. How did they get the ampicillin resistance gene in there? How did the scientists construct this plasmid so that, you know, this P-glow or this gene, that allows for fluorescence, gets present in the plasmid? Because most bacteria you go around looking at, most don't naturally fluoresce. Okay? And we can do this with firefly luciferase. That's the gene that allows fireflies to, you know, burst and show light. Okay? That's basically what you're looking at here. All right? So if we were to, you know, we can move DNA in and out, shuttle it in, and engineer different plasmids for whatever purposes we want to look at. We want to look at, you know, glowing bacteria. Okay? This is called recombinant DNA. Why is it recombinant? Recombinant refers to sort of something that's put back together. It's recombined. Okay? So you have pieces of DNA from different origins that you put back together and make some Frankenstein thing. This is your plasmid. That's the basis of genetic engineering and recombinant DNA. Okay? So this is sort of what occurred. All right? You have bacteria have naturally occurring plasmids. You have this plasmid. You take it out. You isolate plasmid DNA from bacteria, and then you can break down whatever parts of that plasmid you want. Say you want to look at the gene for the fact that jellyfish glow, and you want to make mammalian cells glow and express this outside jellyfish GFP, what we call a green fluorescent protein. Okay? So to do that, you can't just, mammalian cells don't just pick up pieces of DNA. They will express plasmids, okay, similar to bacteria, but it has to be in a certain form. You can't just take the sequence of a single-stranded piece, a non-circular piece of DNA, and put it into mammalian cells. So to do that, you have to take your gene of interest, okay, and you clone it. You put it together. You make a copy of it, and you put it together inside the plasmid. Put the plasmid in bacteria. You make the bacteria will replicate the plasmids for you, so you have lots of it. You isolate the plasmid DNA. Now you have that DNA, and then you can put it into cells. You put it into plants. Now corn doesn't produce seeds. Okay? You put it into apples. Now apples no longer turn brown when they're exposed to the air, which is what has happened. You put it into, you name it, into rice so that they produce yellow rice instead of white rice. Okay? There's a whole long line list of how researchers have used plasmids to alter alter organisms, whether it be plants, bacteria, humans, or etc. Okay? How many of you know someone who's an insulin-dependent diabetic? Where did their insulin come from? Where does that insulin come from? You have no idea. Where do a lot of drugs that you take come from? This right here. You take the gene for insulin, the human insulin gene, you copy it, you put it into a bacterial plasmid, and that bacteria will express that gene. They will produce that protein. Okay? They make more copies of it, they produce the protein. Pharmaceutical companies grow up big batches of bacteria that will, a batch of bacteria that will fill this room, pellet it down, and take all the protein out of there that's produced. And then there you have insulin that is human insulin that is produced for insulin-dependent diabetics, growth hormone, you name a protein or a hormone that you take as a pill, that is how it is produced in mass quantities. Okay? Production of multiple copies of a single gene is called gene cloning. All right? It's used to make many copies and produce a protein product such as insulin. All right? And questions on gene cloning, genetic engineering in general. Okay? Now we can go back and talk about sort of the procedure of taking the gene. How do we get a sequence of the gene into this circular plasmid? Because it just doesn't magically go in there. There has to be there are some sort of parameters that we have to take into account. Okay? So here we have a circular plasmid, right? And here's our gene of interest we want to put in there. Well, if we structure it in a certain way so that the ends of this gene have certain DNA sequences that are recognized by enzymes, okay, and those enzymes are also present in the plasmid, we can use those enzymes to sort of direct where that gene's going to go and how it's going to be directed, eventually be replicated and produced as protein. Okay, so it's sort of directionality, all right, where this is going to go, providing a map to where the DNA is going to be put in. Okay, so restriction enzymes, they cut these DNA molecules at specific sites. Those sites are based upon the DNA sequence. Okay, and so usually a restriction enzyme makes lots of cuts, right? So you might have restriction enzyme sites that look like this, right? So that they cut at each one of those lines on the plasmid, right? So this restriction enzyme will cut this plasmid into different parts, into, let's see, 1, 2, 3, 4, 5 different parts, based upon size. That's not a good procedure to direct where that gene's going to go. Why? You want this gene to go in this spot right here. How do you know it's not going to go into any of the other restriction sites? You don't, right? So scientists have used this, the production of restriction enzymes and the production of plasmids in a certain way to direct where these restriction sites are, so that you only have these two restriction sites, say, in black, right? So you have a site 1, site 2, right? And that corresponds to site 1 and site 2 on your gene. So now there's only one way that that gene, right, your favorite gene, whatever it might be, right, can go into this plasmid. It can only go into one direction. So now scientists can definitively know this is the way it goes, this is the way it's going to be orientated, this is going to be the protein that's going to be produced. All based upon the structure of the DNA itself. All right, so this is sort of what it looks like. Okay? So you have a restriction enzyme will cut the DNA into what we call sticky ends. Why are they sticky? What's unique about this part right here? About this end and that end?
Matching base pairs.
What's that?
The matching base pairs.
So they have matching base pairs. What's unique about them? Cat.
It's not a clean cut, so you can have a complementary segment into it.
So what do you mean by clean cut?
If there's bases left. So
That's what we call an overhang. Okay? You see the bottom strand here has 4 unpaired bases at the end. Is that the way DNA wants to be? No. They want to match up with their complement somewhere. So if you design what we call the insert, right? Your insert is here, is designed in such a way that it matches the overhang. Right? Its opposite strand matches the overhang from the cut site. So what's it going to do? It's going to match up, provide complementarity, and make a double strand. Now that directs where this insert is going to go in the plasmid. Right? This is what we call directional cloning. All right? So then you only have one possible combination. The insert can only go in one direction. It provides for a definitive site where that insert's going to go, and then you produce a recombinant DNA molecule with your gene of interest in the plasmid. Okay? Pretty straightforward procedure. Yes? I will tell you, it took years and years of effort and work to figure and perfect this. Right? And even so, in the lab, this is not that straightforward. If you ever get to a point where you're doing DNA cloning, you will find ways to screw it up. Just saying. All right? But then, how do we know another procedure that's used to examine sort of how and check to see if cloning works, or it's also been used for other uses, okay, is a process called gel electrophoresis. And gel electrophoresis is just a way of separating out DNA based upon its size and charge. Okay, and you're going to do this week in lab. Okay? So you take a mixture of DNA, all right, whatever it might be. Say you start with a plasmid and you use the restriction enzyme to cut it in 4 places. Right? So now it's going to end up into fragments that are, some are bigger. Let's say this is 1, or say that's 4, 1, 2, 3. Right? So 4, maybe 3 is a little bit smaller, 2 is a little bit smaller than that, and 1's the smallest. Right? That's sort of the size, the parts that are going to result from that genetic, that restriction enzyme digest, as it's called. All right? But on how do you know that your restriction enzyme worked? You don't until you check it on the gel. All right? So you use a gel. It's like sort of a semi-solid thing, right? And you load the DNA on a part of the gel, and then you expose that DNA to, or you subject it to electrical current. DNA is charged, okay? It's negatively charged, so it will always move towards the red or the positive end, okay? And so based upon that, you can then move it through the gel, which kind of separates it so it looks like this. All right, so here's the procedure. Here's your mixture of your DNA molecules, right? And you put them into the wells within the gel. Now you subject that gel to electrical current, and the DNA will move towards the red. Right? Always run towards red. The purpose of the gel is to provide sort of a resistance. Okay? Smaller the DNA, less resistance that it has, or the more ability it has to run through the gel, to move through. Okay? The bigger the DNA, the more resistance it has and the less ability it has to run through the gel. All right? Smaller DNA, the further it runs. All right? And this way you end up with something that looks like the bottom picture. All right? Bigger DNA pieces are closer to the wells. Smaller fragments are further away. All right? So it goes sort of biggest to smallest. So what you see there is DNA that is stained with a DNA stain called lithium bromide or whatever. You're not going to use lithium bromide because it's a carcinogen, because it reacts with DNA, obviously, right? And there's other, where I just get a Words with Friends update. This allows for visualization, just like you did for the P-glow. You're going to check your colonies and see if they glowed under UV light. You'll check DNA, see if it glows under UV light with the presence of this stain. All right, so we went over sticky ends. And then DNA ligase is used in normal cells to link DNA. You can use DNA ligase to link DNA in the test tube too. Okay? And then you could say the original plasmid is called a cloning vector. It's a plasmid that is used to put genes in. Okay, so it's sort of the basic structure behind it, and then you add things to it. All right, and that can carry your foreign DNA, the outside your favorite gene, you know, GFP, whatever it might be, that then is added to the vector, the original plasmid. All right? Questions on that? Questions on DNA cloning, gene cloning? All right. So the other big piece, or the other procedure you're going to use this week in lab, is a process called polymerase chain reaction, or PCR. Okay? PCR is a way of utilizing other enzymes in a certain procedure, all right, that will copy a piece of DNA. Okay? So what enzymes do you definitively need to copy DNA? Which enzyme in normal cells synthesizes DNA off the template? DNA polymerase. Okay? This is why this is called polymerase chain reaction. Because DNA polymerase will recognize a single strand of DNA, find the 5' or the 3' OH, and then synthesize a new strand. Okay? So basically you could do this in rounds and rounds and rounds over time, and the polymerase will continue to synthesize a new strand. And we'll go over this in a second. The key to this procedure is a unique DNA polymerase that doesn't disrupt under high temperature. Okay? And it's called Taq polymerase. And then since this was found years ago, sort of it's been perfected and sort of mutated in lots of different ways. All right? The guy who originated this procedure, his name was Kary Mullis. And the story is that he came up with this on a trip to Yellowstone or a national park of some sort. And he went to national park and he saw this geyser, right? A hot spring. And it was found that bacteria live in this hot spring, that it's always boiling. Well, how did that work? He sort of came up with it, and he figured out that he was a postdoc at the time studying DNA replication, and he came up with the idea that bacteria still have to replicate DNA, so how is their polymerase different than others? And can it be used in a sequential fashion to produce lots of DNA? He was sort of a hippie in a way. We'll just say that. A little bit of a hippie. We'll just leave it at that. And he came up with these great, awesome, brilliant ideas. I don't know where they came from. So this is sort of what it looks like. This is the procedure. So here you have a specific gene. Let's use the insulin gene of humans. Okay? We know that all your cells have the same copy of DNA. So your skin cell has DNA that codes for insulin. Okay? You know the sequence of that insulin gene from the DNA sequencing, the Human Genome Project. Okay? So you know that it's ATCG, blah, 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 1,000 base pairs later. If you have a certain part of a primer, right, because you need DNA polymerase needs a little primer to start, if you can produce a primer that also is unique to that gene, it has 20 to 35 base pairs of that gene, of the insulin gene. Okay? I isolate DNA from my skin cells, and I incubate it, put it together with that primer that I've produced. That is unique to the insulin gene. All right? Now, what do I have to do from there? So right now I have a double-stranded portion of my DNA with a primer that's unique to the insulin gene. Well, this is the first step. It's called denaturation. I have to separate those two strands from the original DNA. I separate the strands. Now, what do those strands want to do? They want to come back to one another. You have two separate complementary strands. They want to come back to one another, right? So I separate them out by allowing them to come up to a high temperature. They separate, but before the two big strands come back together, what's going to get in the way? Your primers. The primer is a little shorter, so it will attach to its complementary base pairs before the whole big strand. Okay? So those two strands, they come together, and now that primer leaves a free 3' OH. That is now the starting point for DNA polymerase to start. And now DNA polymerase will recognize that and make a copy of the original template. All right? So first you have denaturation, then annealing, temperature so that at a high enough point so that the whole piece doesn't re-anneal, but the primers do. They attach. And then you get extension. The polymerase will synthesize that whole piece of DNA. Now so for one cycle, through that process, you've created two copies of the original DNA that you started with. Okay? From there, I can go back to the denaturation part. I go back from this point, go back to here. Okay? Then I cycle through and I keep going. All right? So cycle 2, right? So cycle 1 yields 2 molecules. Cycle 2, 4, 8, 16, blah, blah. That's exponential increase. So at the end of 40 cycles, you have an abundance of that piece of DNA that you're looking at the insulin gene that we were talking about. Now you can use that for whatever purpose that you want. This also is the basis for testing, DNA testing, to see if crime scene investigations. Okay? a piece of hair. How do you know that DNA from that person is there? Take a piece of the hair, you isolate the DNA, you do the PCR. Okay? You're making a unique replica of that person's DNA. All right? All right? So it's used, PCR is used to provide the specific DNA fragment to be cloned. So we're talking about making a piece of the insulin gene that can be cloned into a plasmid and gone from there, right? And there's lots of different ways that you can direct and use PCR to incorporate restriction sites. You can use PCR to incorporate changes in the DNA sequence. Say you want to test to see a function of the protein, but you want to see if there's an important mutation in that protein. So you change a couple of the DNA bases. That changes the protein, whether it's an amino acid. That changes the protein, and then you can test to see how that works. That's called mutagenesis. You're introducing a mutation and changing things. All right, and then so this is sort of the cloning procedure, right? PCR, cloning into a recombinant plasmid, and go. All right, and the last thing you can do is DNA sequencing. Right? Ways that you can actually utilize PCR to tell what sequence the DNA is, all right, that you want to. And this is the basis for the Human Genome Project, which most of you grew up with. I mean, it started, the Human Genome Project started probably, I don't know, a few years after most of you were born. Okay? And then there's what we call next generation techniques that make it more rapid, more inexpensive, so that we could do it, say, in the lab at some point. That's the next step where we're going, as it might be. All right? Questions on that? Sarah?
So is the only difference from the PCR that the primer attaches? I'm kind of confused still on the PCR.
So the difference in terms of, so PCR is basically a stripped down DNA replication.
Okay.
All right? You're looking at, instead of the full genome, you're looking at one piece, blunt end, and you want to replicate that piece. A primer sticks in here, a primer sticks in there. Instead of the RNA primer that's used for DNA replication, it's a DNA primer, okay? Still provides the 3' OH, and it's just unique to whatever gene you're looking at. So now you're only replicating this part, that part goes together. There's no other primers so the only replication the only spot that DNA polymerase can start from is the end of that primer. So you're only looking at that particular spot, all right? So it's a stripped down DNA replication, so it only happens at your specific area of interest. Emily.
What happens in the annealing stage again?
So the annealing stage is where denaturation occurs. You heat all this DNA up, and the DNA separates because the two strands are held together by what? What holds the two strands of DNA together? Hydrogen bonds. So if I have hydrogen bonds that are holding them together, I subject those hydrogen bonds to heat that breaks the hydrogen bonds, and the strands fall apart. Right? Not so much heat that the original DNA template those covalent bonds are gonna break, but enough to separate the hydrogen bonds. Now I slowly lower the temperature. Okay? As I lower the temperature, what's going to start to form? Hydrogen bonds. That's where annealing is. You're producing those hydrogen bonds between the primer and the template DNA, okay, by lowering the temperature in a slow fashion, so that the primers anneal and not the full template to template. Okay? Questions on that? So this week in lab, you're going to do PCR. All right, you'll have specific primers. You're going to do PCR, and you're going to do gel electrophoresis, I think. or next week in lab you do PCR. I don't know. I know you're doing either restriction enzyme digest and gel electrophoresis 1 week and PCR the other. Okay. Other questions on PCR? And you go over this again in lab. Sort of look at the specifics of what you're doing. All right. Know that this is a technique that allows scientists to do lots and lots of different things. All right. All right. I'll see you guys on Wednesday.